Hi, my name is Karen Johnson, and I am a teacher at Evanston Township High School. I teach a couple different levels of introductory biology, and the unit that I worked on this summer is about the central dogma, so that's the DNA codes for RNA, which codes for protein. This unit has been a struggle for a lot of students in the past because of how technical DNA and RNA seem when you jump right into them. So we're approaching it from a slightly different angle. The first thing the students are going to see is a video of these two young women who are twins, and as you can see, one of them is black and the other one is white. So we're going to try to figure out why it is that they look different. So framing this whole unit as sort of a mystery of figuring that out, I hope will increase student buy-in. We're going to focus specifically on their hair because hair is a non-invasive, easy thing to test. So we're going to do some biochemical analysis of hair and see that it is made of protein. And under the microscope, you can actually see the pigment that's within the hair also. So we're going to look again at the pigment there. Using this computational model that allows us to look at people with different hair colors and check the pigment levels for each hair color and record that in a table and graph so we can look at different hair colors, check out the different protein levels within those hair, and see what the relationship is between those pigments and the hair color. We're then going to do an overview of proteins and look specifically at the structure-function relationship of proteins and how important that is. And this lesson is going to be using more traditional means of videos and readings. But then we're going to need to figure out how that protein gets made. So we're going to jump at that point into DNA and RNA and use this sort of test tube model where students are going to be able to add different components of things to either make new DNA through DNA replication or make RNA through transcription. So here, let's make some DNA. So we're going to need some deoxyribonucleotides and DNA polymerase. And the students are going to be able to see the random motion of nucleotides and how they need to be in the right place at the right time for the enzyme DNA polymerase to be able to incorporate them into the growing DNA chain. They're also going to see as the new strand gets built, they're going to see Chargaff's rules emerge here. So they're going to be able to see that the percent A and T are going to be equal and the percent C and G are going to be equal across the old strand and new strand. There's also a fun bug in this system, so students are going to have to use actual biological content knowledge to fix a bug in this, which I think is going to be fun for them to do. Then we're going to need to figure out how protein gets made based on the instructions from DNA and RNA. So we're going to use a model set up by the Concord Consortium to watch an animation of the process of translation, so protein synthesis. And we're going to ask students to use this and traditional tools like codon tables to figure out how the amino acid sequence that gets incorporated into the nucleotide or into the new proteins is dictated by the nucleotides from the DNA and RNA. Then we're going to tie it all together at the end using the actual or part of the actual gene that codes for red or brown hair. So this version of it, it's already here, this version of it would code for brown hair. But there are some specific nucleotides represented in this sequence that, when mutated in real people, would lead to red hair. So this is a similar model to what we saw before with our replication and transcription model. But instead here, we're going to have our template strand, and students can actually manually change this or input their own nucleotide sequence. And we need some deoxyribonucleotides and some polymerase, and we are also able to change the error rate of our enzyme. So we can look for lots of mistakes or very few mistakes and have replication occur as it was before. And once the gene is done replicating, we'll be able to check this person's hair color. So if those couple of nucleotides are mutated, the person's hair is going to change to red. So students will be able to synthesize the whole unit together and see that the genes themselves, the DNA sequence, impacts the characteristic that we were looking at. So through these models and other experiences, students are going to be able to do a lot of pattern recognition. They're going to be able to design and carry out experiments with these tools. They're also going to be able to play around with some computer models and really compare and contrast the strengths and weaknesses of those models. So through these computational models and more traditional teaching, like videos and some readings and class discussions, they're going to be able to put this difficult concept together in a more organic and impactful way. So thank you very much for watching.